बिटिया, हमेशा खुश रहो बिटिया. Myself, Dr. Arti Purohit, JVL, Professor, Pathology Department, Faculty of Homeopathic Sciences. Today we are going to start a new lecture on difference between the gram-positive and gram-negative cell wall. The bacteria has a unique feature in its cell wall. Only the bacteria word come because of cell wall. So, before the gram staining comes, we know about the bacteria as a bacteria. But now we will classify the bacteria into two parts. One is a gram positive one and second one is a gram negative one. Dr. Christian Gram gives the gram staining technique through which we are able to see the cell wall of the bacteria as a gram positive and gram negative. The major difference between the gram positive and negative cell wall is that the gram positive bacteria has a thickened cell wall, whereas the gram negative one is a thin cell wall. And the gram positive one has a less permeability and gram negative has a more permeable. Gram positive bacteria actually retains the primary stain, that is the crystal violet. It will not decolorize by the acetone and do not take the next staining, that is the safranin. Whether the gram negative one will decolorize by the acetone and again take the next staining in the technique, that is a safranin stain, and it will look under the microscope as a red color gram negative one, while the gram positive, because it retains the primary stainings, that's why it is looking as in blue in color. In gram staining, definitely before that we have to understand the gram staining technique. In gram staining, we will add in the fixed smear, we'll add the crystal violet, then decolorize with the acetone, and again secondary stain, that is the safranin. The bacteria who retains this primary stain, that is a positive, and the bacteria who decolorize by the acetone and again take the next staining, that is a safranin, they are red in color, are the gram negative one, and it is the gram positive one. Now we'll talk about the diagram of the gram positive and gram negative cell wall. Here it is in gram positive cell wall, which contain the peptidoglycan layer, which is thick. Here is a peptidoglycan layer which is around 18 to 20 nanometer, whereas the gram negative one has just the 6 to 7 nanometer. This peptidoglycan is making the gram positive bacterial cell wall as a less permeable, and it is a cell membrane which has a PLP model, as you know, peptide. Here, after the peptidoglycan layer, there is no outer membrane here, while the gram negative cell wall, there is again the PLP model, but here peptidoglycan layer is a thin one and there is an outer membrane, then again PLP model, and here is the outer membrane. There is a lipotecoic acid in it. In the diagram, it seems that gram negative is a thick one, but actually the gram negative layer is the thinnest, and here is the peptidoglycan layer which is the less, which is just an 8 to 10 nanometer, while the peptidoglycan layer in the gram positive is a 18 to 20 diameter, and there is absence of outer membrane. Outer membrane is absent, while outer membrane is present in the gram negative cell wall. Here is a PPT, peptidoglycan layer, and it is the innermost membrane. It is a periplasmic space. Periplasmic space is merged in the gram positive one. Here is there is no periplasmic space or less in the gram negative layer. There is this is the only thing which will tell about the permeability of the cell wall of gram positive and gram negative. Now, after the diagram, you can understand that this peptidoglycan, there is a L, in spite of L-alanine, there is a D-alanine in the gram negative. In this peptidoglycan layer, there is a blocking of bond. L-alanine, D-lysine, and this is an phosphate bond. But in this gram negative, there is no bonding and no that is why it is more permeable. I hope you enjoy this difference between the gram positive and gram negative bacteria. Now you can understand what is the difference between gram positive cell wall and gram negative cell wall. This is all about. Thank you so much. Bye bye.